DNA nanotechnology as a tool for synthetic biology. Some of you may be familiar with synthetic biology as using genetic information to design and manipulate biological systems, like, say, encoding a biosensor. But this doesn't fully explain the scope of synthetic biology. So what is it, and how is it related to DNA nanotechnology? I would say, to cover all my bases, that synthetic biology is the application of engineering principles in the rational design of biological systems and devices. It's also important to specify that SynBio is interested in investigation as well as design. Trying to understand the ridiculously complex biology of the cell is no easy task, with the multitude of biomolecules that undergo biochemical as well as structural interactions in this complex environment. A subset of synthetic biology described as the bottom-up approach suggests that we strip back a cell to the minimal components necessary to life. We can then work towards an understanding of biomolecular interactions in an environment easier to analyze and control. Synthetic biology is more than just genetics. It also involves creating novel, non-native structural molecules that are protein or DNA or even RNA based. So why choose DNA as your structural material? DNA is already biocompatible, and as the blueprint of the cell's genetic information, it's highly stable and replicates with highly specific sequence hybridization of anti-parallel complementary strands using Watson-Crick base pairing between nucleotides. It's able to form higher order structures, and it is also governed by the interaction of its nucleotide bases, while the backbone remains unchanged. Thus, it is relatively simple to use these governing properties to make designs. Biological membranes are a particularly relevant application for DNA technology in synthetic biology, as these fundamental partitions are governed by membrane proteins, which due to their amphipathic properties are extremely hard to isolate and study in the native state, which is an issue in modelling and manipulating membranes, as over 1/3 of the proteome is dedicated to these membrane proteins. The phospholipid bilayer is composed of hydrophilic phosphorus heads and hydrophobic fatty acid tails in a fluid mosaic form that forms a semi-permeable partition which allows biomolecules to be separated in space for specialized functions. When creating a DNA nanostructure for manipulating or modeling membranes, there are multiple components that can be designed in order to achieve desired functions. Binding of DNA can be finessed by changing parameters such as ionic strength, concentration of DNA, and temperature externally. The nucleic acid is water-soluble, can be designed to match nucleotide recognition particles, or structurally specialized to interact with the membrane. For example, act as a gated nanopore or an ion channel. In order to incorporate the DNA nanoparticle into the lipid membrane in order to perform its function, a neutral linker such as polyethylene glycol can be used to attach a molecule with lipophilic properties, for example, cholesterol. It is also possible to attach a reporter molecule in order to track the progress. Using DNA, it's possible to create precise structures that are accurate to the sub-nanometer level, with biochemical interactions specialized by the linked species you attach. Furthermore, higher order structures such as DNA brick and DNA origami are possible. Not that kind of origami. The first person to pioneer the idea of DNA as a structural material was Samani, who, as a crystallographer, hypothesized that DNA could be used as a structural lattice to keep molecules in a regular array. Nowadays, there are complex arrangements of DNA nanoparticles. Notably, DNA nanoparticles with lipophilic properties have been shown to be able to stabilize lipid nanotubes, which are able to form structural and chemical connections between different vesicles or synthetic cells. Nanoparticles are also capable of mediating vesicle transport by recognizing specific nucleotides. DNA nanotechnology is an extremely useful tool for synthetic biology that could be applied to biotechnology applications, energy, nanomedicine, or even nanorobots. Importantly, the reason DNA nanotechnology is so useful to synthetic biology is because of its simple governing properties that allow rational design